Hello, my name is Rachel Martini, and I'm a postdoc in Melissa Davis's lab at Weill Cornell Medical College. I would like to thank the organizers of this aortic annual meeting for allowing me to discuss the project entitled Transcriptomic Analysis of Triple-Negative Breast Cancer in West versus East African Patients Reveals Distinct Immune Responses. Breast cancer remains a leading cause of mortality among women globally. We and others have shown that the molecularly aggressive breast cancer subtype, triple-negative breast cancer TNBC, is more frequently diagnosed among women with shared West African ancestry. Specifically, looking at prevalence rates among our ICS-BCS cohort, we've reported the highest rates of TNBC among Ghanaian and African American women compared to lower rates among Ethiopian and European American women. When we quantified West African ancestry across our cohort, we also showed that the percentage of West African ancestry was significantly higher among women with TNBC compared to those with a non-TNBC diagnosis. To further investigate differences among TNBC, we sought to determine if we could identify ancestry-specific drivers of gene expression among these tumors. We've done so with a preliminary RNA sequencing cohort of triple-negative breast cancer cases from Ghanaian, Ethiopian, and African American women to specifically see if we can identify an African ancestry-associated gene signature. In our RNA sequencing cohort, we estimated genetic ancestry from our alignments using 1,000 genomes populations as our reference. We observe a range of African and European admixture across our cohort, with Ghanaians having the highest proportion of African ancestry, followed by African Americans and Ethiopians. Interestingly, Ethiopian women with TNBC in our cohort reported similar levels of African and European ancestry, where European ancestry was predominantly represented by Italian ancestry. We also observed Asian admixture among our Ethiopian patients. To identify differentially expressed genes, we utilized both ancestry as a continuous variable and self-reported race ethnicity as a categorical variable to identify ancestry-specific and self-reported race and ethnicity-specific gene signatures. We did observe some overlapping genes between these approaches, but each analysis revealed gene signatures that were either ancestry or race ethnicity specific. Unsupervised hierarchical clustering of our African-associated gene signature separated our samples into two main nodes, shown here as low and high AFR cluster, where low AFR cluster represents predominantly Ethiopian samples in our cohort, and high AFR represents Ghanaian and African Americans. To look for pathway enrichments in our African ancestry-associated gene signature, we calculated log fold change between the clusters and utilized ingenuity pathway analysis. Ingenuity pathway analysis of the ancestry-specific gene signature revealed an enrichment in terms related to immune cell trafficking, where terms related to migration and movement were predicted to be activated, and terms associated with immune cell activation were inhibited in those with higher African ancestry. Using CyberSort X deconvolution, tumor-associated leukocyte abundance was significantly higher among high AFR samples, and this seems to be driven by increases in B-cell, T-cell, and monocyte populations. 751 genes were unique to our self-reported race-ethnicity gene signature, and unsupervised clustering of these samples revealed that individuals separated more by geographical location versus ancestry, where African Americans clustered distinctly from Ghanaians and Ethiopians. Pathway analysis revealed enrichment in a number of potentially comorbid-related pathways, such as cardiac signaling pathways, diabetes-related pathways, and pathways related to obesity, which were predicted to be activated among African Americans. In conclusion, use of both ancestry and self-reported race ethnicity-based analysis approaches allowed us to identify distinct gene signatures and pathway enrichments, and highlighted the importance of considering both an individual's genetic ancestry and self-reported race and ethnicity in the context of cancer genomics. I would like to briefly thank all of our collaborators for their contribution and support of this work, as well as our funders. Please contact me for any questions you may have. Thank you for your attention.